Hello and welcome back to Immunology, The War is Over, Episode 7, The Complement Cascade. When I first pictured this series in my mind, I set aside only one episode for complement and disease. But when I actually sat down to create it, I quickly realised that one episode was not going to be enough. And so in this episode, we're going to take a deep dive into the complement cascade. But then we'll follow on from there with some additional episodes which will focus on the complement regulators and some complement-mediated conditions. So stay tuned for those. Okay, so the complement cascade. In this episode, I'm going to present complement to you in two ways. Firstly, we'll just recap on the basics, and then we'll do a deep dive. So depending on what you need for your studies, the basics might be enough for you and you might want to stop there. But I also wanted to present the deep dive version because complement is an area where the literature is exploding. And the role of complement in various disease states is expanding across multiple specialties. So as a doctor, regardless of which field you're in, understanding complement is likely to come in handy in your future practice. So take what you need from this episode. If basic is enough, stop there. If you want a deep dive, then keep going until the end. And what's super fun about this episode is it's going to be handwritten. I've provided you with a handy worksheet which you can download, or you can just grab some blank paper and some colourful pens or highlighters and you can scribble along with me as we go. So grab what you need and let's jump right in. So let's start with the basic overview, which we touched on back in episode 5. Complement is an incredible part of our innate immune system. It's a proteolytic cascade which generates opsonins to increase phagocytosis, anaphylatoxins to attract immune cells to the area, and the membrane attack complex which punches holes in target cells, causing them to die. And this goal of complement can be achieved via 3 different pathways: the classical pathway, the lectin pathway, and the alternative pathway. The classical pathway is predominantly triggered by immune complexes, so an antibody stuck to the surface of a pathogen could spark this off. But equally, the classical pathway can be triggered by damaged host tissue as well. And the lectin pathway is pretty similar to the classical pathway, but it's triggered slightly differently, being sparked off by carbohydrate sequences on the surface of pathogens, or by those same sequences on damaged cells. And finally, the alternative pathway, which is the sassiest of the pathways. This is always ticking over in the background and will activate quickly when it comes into contact with foreign cell surfaces. And whilst these pathways are described separately, they're not entirely exclusive. They each have a tendency to feed into and amplify each other. But regardless of which pathway is sparked off first, these 3 triggering pathways converge on the production of C3 convertase, then C5 convertase, and finally the formation of the membrane attack complex, which punches holes in cells, causing them to die. And along the way, the cascade produces anaphylatoxins and opsonins. Anaphylatoxins, C3a and C5a, attract immune cells to the area and then promote the function of those immune cells when they arrive. Opsonins, such as C3b and inactivated C3b, promote phagocytosis of the cell they are attached to. And there are some drugs which have been designed to interrupt this cascade. Top of the list is eculizumab. Eculizumab is a monoclonal antibody which blocks C5 and so blocks the terminal pathway of complement. This drug is one of the most expensive drugs in the world and is primarily used to treat 2 rare conditions for which there are no other effective treatments available, namely atypical HUS and paroxysmal nocturnal hemoglobinuria, or PNH. Another complement blocking drug that's shown up in clinical trials recently is the oral C5 inhibitor avacopan. This has recently been trialled as a steroid-sparing agent in ANCA vasculitis. Okay, so that's the basic overview done. If your complement cup is now full and you want to stop there, that is absolutely fine. But be sure to tune in again soon for the bonus episodes where complement and disease come together. I think you'll find them really useful for your exams. Now, for those of you who want to stay for the complement deep dive, get ready to draw out the most beautiful complement cascade you have ever seen. As we draw this out, we'll be building on the flowchart diagram that we just made, but we're also going to place these complement molecules on the cell surface, so you can visualize what is actually happening and where it is happening, so we can demystify this little cascade once and for all. So let's start by unpacking the classical pathway. This is triggered by immune complexes, but it also interacts with dead cells and acute phase proteins. To understand this pathway, we must first be introduced to the C1 complex. The C1 complex is composed of C1q, R, and S. Isn't it delightful when the alphabet is used to keep track of things? So these molecules come together to make the C1 complex, and it almost looks like a little bunch of flowers. The C1q molecules form the stems and the flower heads, and the C1r and C1s molecules decorate the base of the bouquet. But this is not a benign bunch of flowers. It's a potent little pattern recognition molecule, and I'm pretty sure it would feel moderately patronised at being likened to a bunch of flowers. Anyway, these flower heads of the C1 complex are binding sites which recognise IgG and IgM immune complexes, as well as exposed structures on damaged cells. When C1q binds to something, it cleaves and activates C1r, which in turn cleaves and activates C1s. And C1s will now spark off the entire complement cascade by cleaving C2 and C4. Now, something you'll notice as we move through this tutorial is that as complement factors are cleaved, they divide into two parts, part A and part B. Again, solid use of the alphabet here. As a general rule, part B wants to bind and belong to a team, whilst part A would rather be alone. You'll see why I'm telling you this in a second. So C2 and C4 are cleaved into C2A, C2B, C4A, and C4B. C4B and C2B want to belong to a team, so they buddy up with each other on the cell surface, and their A counterparts fly away. I must say I couldn't find anything in the literature which assigned a particular function to C2A or C4A molecules, so we'll just call them byproducts for now. But C4B and C2B team up to make a bigger molecule, C4B, C2B, which is a C3 convertase. Now, just a little side note, historically older resources denote this as C4B, C2A. But the current modern terminology is C4B2B, which, let's face it, is much easier to work with because we have our little rule that part B wants to bind and belong to a team. Once this C3 convertase establishes itself on the cell surface, we are in business. C3 convertase now cleaves C3 into C3A and C3B. C3A is an anaphylatoxin, which will now go off and promote inflammation. But C3B again wants to bind and belong. So C3B will now join forces with the C3 convertase molecule, C4B2B, to become C4B2B 3B, which is otherwise known as a C5 convertase. And C5 convertase, as you would imagine, cleaves C5 into, you guessed it, C5A and C5B. C5A fulfills its role as an anaphylatoxin, and C5B, again, more of a team player, joins up with C6, 7, 8, and 9 to form the membrane attack complex. Easy! Okay, so that's the classical pathway done and dusted. Now let's shift our attention to the lectin pathway, which is actually very similar to the classical pathway, but with a few new immune characters in the mix. Now, I don't know about you, but the lectin pathway was something I misunderstood right from the start. I knew there were molecules on the surface of microbes which sparked everything off, but I didn't understand the spark itself. And the spark is fascinating. The spark begins with something known as lectins. Lectins are pattern recognition molecules which bind to particular carbohydrate groups, and when they do this, they spark complement. And the carbohydrate groups that are recognised by lectins are not only present on bacteria, but also on viruses and fungi. So lectins have a broad reach when it comes to identifying pathogens. And whilst these carbohydrate groups are expressed on the surface of microbes, they're not a feature of healthy human tissue. But in saying that, if we have cell damage, those same carbohydrate groups can be exposed. And so just like the classical pathway, the lectin pathway also has a role in clearing out damaged cells from the body. And we have a few of these lectins whizzing around inside us, the most famous of which is MBL, mannose binding lectin. MBL binds to a variety of bacteria and viruses, including HIV. But interestingly, when it comes to fungal infections, MBL binds to mannan, which is a key component of the fungal cell wall. It's part of what makes a fungus a fungus. And so it goes without saying that MBL has significant antifungal properties. And whilst MBL is by far the most famous lectin we also have plenty of others, such as the collectins and ficcolins. So lectins are pattern recognition molecules which can trigger the complement cascade. But the true spark for complement is actually generated by what those lectins are attached to, something known as MASP, MBL-associated serine protease. As you can imagine, this serine protease is in the business of cleaving proteins. In other words, MASP is the true complement firestarter in the lectin pathway. It sparks everything off. And when we draw this on our diagram, what you'll notice is that this MASP-lectin complex looks very similar to the C1-QRS complex. And indeed, this is another potent little bunch of flowers. We have the little flower heads, otherwise known as carbohydrate recognition domains, which, when they bind, signal downstream to MASP, which will in turn become activated and spark off the whole complement cascade. And MASP does this by cleaving C2 and C4, and so sparks off complement in the exact same way that we just saw in the classical pathway. Now, as with all things in the human body, and particularly the immune system, it's actually more complicated than that. There are different types of MASP and they interact with each other. So MASP1 activates MASP2, which sparks off the lectin pathway we just described. But MASP1 also activates MASP3, which is thought to promote the alternative pathway by activating factor D. And of course, we'll be discussing the alternative pathway shortly. So that's the lectin pathway. Lectins are pattern recognition molecules connected to a potent spark called MASP. When they bind to particular carbohydrate motifs, they will recruit complement to annihilate organisms and/or remove damaged tissue. Okay, so now we have the classical and lectin pathways pretty much sorted. Now let's switch gears and get to know a real sassy part of complement, the alternative pathway. The alternative pathway has sass in spades. It's a law unto itself and requires a significant amount of policing to keep it under control. And in getting to know the alternative pathway, we must first become acquainted with some brand new characters. Introducing factor B, factor D, and properdin. These are exclusive to the alternative pathway. And for the purpose of this tutorial, we're going to use some wordplay to help us remember what these molecules actually do. So, B is for binding, D is for dividing, and P, properdin, is for promotion. And this will make a lot more sense in a tick when we see these characters in action. But before we do that, I just wanted to highlight another unique feature of the alternative pathway that gives it particular sass and sets it apart from the other pathways. And this is the fact that the alternative pathway is always ticking over in the background, ready for action. So unlike the classical and lectin pathways, which rely on specific pattern recognition molecules to get things going, the alternative pathway does not require such a molecule and can be spontaneously activated of its own accord. And in fact, this is happening continuously at low levels all the time, the so-called tick-over mechanism. But this is confusing, right? What on earth do we mean when we say the alternative pathway is ticking over in the background? Let's unpack that now. When we say the alternative pathway is ticking over, what we really mean is that C3 is spontaneously and continuously dividing into C3A and C3B. And all it needs to do this is water. So C3, in the presence of water, happily divides into C3A and C3 watery B. Now, these molecules are short-lived, so they're always ticking over, but there's not a lot of them around in any one moment. But under the right circumstances, these do have the potential to spark off the entire complement cascade. So let's check out the alternative pathway in all of its cascading glory, starting with the spontaneously generated C3 watery B. This binds to factor B in our plasma, and B is for binding. When factor B binds to activated C3, factor D comes along and divides it. D is for divide. Factor D divides or cleaves factor B into BA and BB. BA flies away whilst BB remains bound to C3B, generating C3B BB, which is a C3 convertase capable of cleaving and activating C3. Now, this C3 convertase is referred to as the fluid phase C3 convertase because it's generated in the plasma, but it will then try to establish itself on the surface of cells. In this initial form, it's actually pretty unstable and prone to breakdown by complement regulators, but this molecular complex can be strengthened by properdin. P is for promotion, and properdin binds to this molecule to become C3b BBP, which is a much more stable version of C3 convertase, which resists breakdown by complement regulators and so remains active on the cell surface for longer. So the plot thickens. You can see now that we actually have two different versions of C3 convertase, depending on whether you start the cascade with the lectin, classical, or alternative pathways. And similarly, there are two different versions of C5 convertase. In the alternative pathway, another C3b is added to C3 convertase to make C5 convertase, like so. And so what becomes clear is the importance of C3b. C3b is a part of the C3 convertase in the alternative pathway, but it's also a key part of C5 convertase in both pathways. And so C3b is a key component of the complement cascade at large, but it also helps to establish a positive feedback loop. Whenever a C3 convertase is generated, it makes more C3b, which in turn makes more C3 convertase, and so on and so forth. But not only do these molecules increase the production of each other, they can also be added together to form C5 convertase, which will in turn lead to the formation of C5a and the membrane attack complex. And so this cycle of C3b and C3 convertase promoting each other, and indeed the rest of the cascade, is known as the amplification loop. And whilst the amplification loop technically belongs to the alternative pathway, it will be sparked by the classical and lectin pathways too. So every time complement is activated, you can expect to see this amplification loop in motion. And this amplification loop is potent, sort of like adding gasoline to a burning flame. You'd want to be careful not to add too much or things could quickly get out of hand. And the same is true for complement. We'll see in the next episode that there are a number of complement regulators, many of whom make it their mission to interrupt this amplification loop by deactivating C3b and destabilizing C3 convertase. So ultimately, the amplification loop is the balance between two competing cycles, the formation of C3b and C3 convertase, as well as the breakdown of these molecules. Now, we said before that the alternative pathway doesn't have a particular trigger or pattern recognition molecule, and it's always ticking over in the background spontaneously. But that doesn't make perfect sense either. Sure, it's always there in the background at low levels, but how does it then ramp up to the full-blown cascade? And this ultimately comes down to C3b binding to a surface it can thrive on. So C3b is always being generated in the background, and C3b is an opsonin which will bind to the surface of cells in its nearby vicinity. So when C3b is produced, it will absolutely bind to nearby cells, including healthy cells. But our body cells are fully aware of this issue, and they use complement regulators on their surface to deactivate this molecule in a jiffy. However, if C3b happens to land on the surface of a bacterial cell, which doesn't have any complement regulators, it will be allowed to establish itself and thrive. And this becomes the spark for the alternative pathway. When C3b is allowed to thrive on the cell surface, it will recruit all of its buddies from the alternative pathway. So factor B, and properdin, and in doing so generate C3 convertase and set up the amplification loop that we saw before, which in turn will spark off a full-blown complement attack on that cell. So that's the main way that we go from the alternative pathway ticking over in the background to sparking the complement cascade. And you'd have to agree, it's pretty clever. But that's not the only way the alternative pathway can be activated. And whilst technically pattern recognition molecules are not essential for the alternative pathway to happen, there are a few molecules which can help things along. For example, properdin, when it binds to a microbial surface, can actually recruit complement to that location. So, as well as being able to stabilise C3 convertase on the cell surface, it may actually spark things off in the first place. Another molecule which has the potential to do this is P-selectin, which is secreted by endothelial cells and platelets. And the magic of the alternative pathway doesn't stop there. Get this, properdin is not made in the liver. It's actually made inside our white blood cells. Monocytes, macrophages, and T cells can secrete properdin into our bloodstream. But other white cells, such as neutrophils and mast cells, actually store properdin in their granules. And this is where it gets really good. The complement cascade generates C5a, an anaphylatoxin, which attracts neutrophils to the area. When the neutrophils arrive, they release their granules, which just happen to contain properdin, which we now know can spark complement from the cell surface and can also stabilise C3 convertase, thereby promoting that amplification loop, which will make more C5a, more neutrophils will come to the area, spew out properdin, and so on and so forth. Isn't that incredible? And it's just one of the many examples of teamwork that takes place inside our immune system. And I won't go into detail on this, but it's important to acknowledge that complement doesn't just intermingle with our innate immune cells, it also influences our adaptive immune system as well, our B cells and T cells. For example, B cells have a particular complement receptor on their surface, CR2, which sits very close to the B cell receptor. When a B cell comes across its matching antigen, it will scan the nearby surface for complement using this receptor. If a B cell finds complement together with its antigen, it will lower the threshold for activation of that B cell. So when complement is there, you'll need fewer antigens to get that B cell excited and present that antigen to T cells. So that was the complement cascade in all its glory. And now we have this fabulous diagram which summarises the whole thing. Now, whatever you do, hold on to this diagram because you're going to need it for the next episode when we see those complement regulators in action. I'll see you in there. Bye.